Hello, good morning everyone. I hope everyone's feeling well today and enjoyed the festivities last night. I'd like to say thank you very much to PacBio for letting me come here today and give you an update from the Tree of Life project. Okay, so for anyone who's not familiar with the Tree of Life program, we are a program at the Wellcome Sanger Institute based in Cambridge. And we are involved in several major projects, all of which are aiming to assemble reference-level genome assemblies for eukaryotic species. So I'm here today speaking on behalf of non-model organisms. So our biggest project at the moment is the Darwin Tree of Life project. We're aiming to sequence 70,000 species from Britain and Ireland. We're also involved in the aquatic symbiosis genomics project. That's even more ambitious. These are all species from marine environments, and we're looking to sequence host and symbiont in one sample in one go, and then bioinformatically disentangle all of those data and assemble the genomes. We're also a collaborator in the Vertebrate Genomes Project, so that's the one that's been ongoing for the longest. We are also a partner within ERGA, the European Reference Genome Atlas, and I know we have some other members in the audience today. And all of these projects are under the Earth BioGenome Project, so they all fall under that umbrella. And this project has the amazing ambition to sequence all life for the future of life. So here we're really looking to fill that reference library. But the main thing that I would like you to take away from this slide is that we have an enormous scope. So we're looking at anything eukaryotic, from fungi to arthropods, anything that you see represented in our tree here. So in order to do that, we've assembled our genome engine, and this is the path that samples take to go through that process. So we begin right at the beginning, here, with the sample acquisition. So particularly for the Darwin Tree of Life project, we have a number of partners all around the UK who collect specimens and send them through to us. They go through COPO, which is a system that ensures that all of the taxonomic IDs are lined up with the different repositories. They are sequenced for DNA barcoding to ensure identity meets up with the taxonomic ID from the experts. And then they start their journey really through the Sanger Institute. So they come through to my laboratory for DNA and RNA extraction. They go through to our colleagues in scientific operations, and we've got James here who leads that team, who do the library prep and sequencing. All of that sequence data is then assembled on our Tree of Life QC portal, so it brings all of the data from the different pieces together, and our assembly team then make a primary assembly. They scaffold with Hi-C, go through to curation, and then everything is open access as soon as possible. So as soon as the sequencing runs have gone through QC, anybody can log on to Tree of Life QC, they can look up that species, and they can see how that species has fared, what kind of data have been revealed. Everything goes onto the open data portal. And the final part here is the publication of a genome note. So this is a very small publication for each species where the genome has been assembled and released. So what have we done so far? There could be some useful numbers on here for the competition. So we have sequenced about 5,500 species. They are in progress at the moment. 161 terabase of PacBio data so far to date, just for this for the Tree of Life program. An awful lot of Hi-C data. We've got 2,316 species in assembly. That's my favorite number because that means that my team, our work is done. We've generated all of the data. It's now with the assembly team and the bioinformaticians to bring it all together. Curation has been scaling massively. 140 manual interventions per gig of assembly. There's a lot of work and a lot of real expertise in that curation team. And then in terms of public releases, we're up at 1,471 assemblies that have now been released to INSTC, and the genome note publications do kind of follow a little bit after that. So at the moment we're at 693. So these numbers are from around 2 or 3 weeks ago. And what you can see represented here is how those species actually play out around the broad breadth of diversity. So you can see the color coding, which tells you which taxonomic area these species are in. And then the red marks around the outside show you completed genomes. And you can see it's a pretty even spread. They go all the way around. Even more even are the blue, which are in progress. So these are species that might have some PacBio data, but we need to do more sequencing to reach coverage, or perhaps we are waiting for the Hi-C library to come through. But they've started their journey. They're in-house. They are being worked on. The red scale around the outside is actually a logarithmic scale, and that's because here are the Lepidoptera. Now, if that was a normal scale, then that red line would go somewhere beneath the floor, because we've done hundreds and hundreds of Lepidoptera. They just happen to be exactly the right size to be dissected and go through our pipelines really well. But I'm here today to give you our take on what's really required in the wet lab, kind of upstream, in order to get this number of species through. So when you boil things really, really down to the basics, you need to homogenize your sample, you need your high molecular weight DNA extraction, fragmentation, you need to clean this up, and you need to do an RNA extraction. And then all of those different preps need to be sequenced and put together at the other end. So how do we do this in the Tree of Life? Well, we've developed a suite of standardized protocols, which is developing and evolving all of the time. We tried to be as open access as we possibly can, so all of these protocols here are published on protocols.io, and the QR code will take you there. And what this means is that we've developed a kind of modular approach, which means that we can really progress at pace. We can have routine QC, the same metrics that are collected at the same points for all of our samples going through. And it means that at the same time as publishing genomes, we're also learning from that data set as it goes through. So we can learn and evolve at the same time. So I'll take you through each of these steps in a little bit more detail and describe what the protocols entail. So the first stage is the sample homogenization, but initially, actually, we need to do sample prep and triage. So all of the samples, although they've been collected by very dedicated professionals, we need to first take a look at them, see what kind of size they are, what's possible, what's achievable. So we have quite a complicated decision tree that's in that triage and dissection prep there. And then we have three main methods for homogenization that are currently in use: the power masher, which is on the far left, the cryoprep from Cavaris, which is your sample goes into this bag here and is smashed at cryogenic temperatures to pulverize what was in there, I think, a fly. And then we also most recently brought the FastPrep 96 online. This has been really important for us. The use of bead beating has actually increased our yield in a lot of areas for the DNA extractions. And although we were initially concerned about the integrity of the high molecular weight DNA, we found that that is not an issue. So anybody who's considering that, I would fully endorse. We do need to do this at cryogenic temperatures, though. Okay, after homogenization, we have the actual DNA extraction. So as you can see, there's a lot more protocols here. The ones that are in bold are ones that are currently in use, and the ones that are slightly faded are previous ones that we've since deprecated. We actually have more that we need to add onto here, and you do need lots of different protocols, and you need to do quite a lot of trial and error to make sure that you've got the right protocol for the right specimen. Two things that I would draw to your attention here are that for a lot of these, we have a manual version and an automated version. A lot of our automation is on the Kingfisher Apex. We found that to be really, really useful and very flexible as well, so that's been fantastic. And I'm sure our colleagues outside from Agilent will be pleased to see this femto trace here. These, for us, are extremely important. You'll be seeing a lot more femto traces, so I hope that's of interest for everybody. Okay, then we have our two main fragmentation protocols. We have the Megaruptor on the left there, which we use upstream of low-input library prep. And we actually use the G-tube upstream of the ULI library prep. Find it a bit faster and a bit more versatile. We then have the spry, which again we have an automated or a manual version of that, and the automation is in the Kingfisher. And we do RNA extractions. For a long time we did manual trisol, and now we've moved that onto the Apex with an automated Nirvana method. We find them equally good. We have a really high success rates for RNA extraction and RNA-seq. So how does this then fit together? Well, for example, if you have a moth sample, this is the path that your moth will take through our processes. So moths will usually be power mashed. They'll go through the automated magatract extraction. Normally, we have enough DNA to go for an ally library prep. We can automate the spry, and we'll do the Nirvana RNA extraction. For a grass sample, we would go for bead beating, the automated plant magatract, because we find grasses are actually quite easy to work with these days. And again, the yield is normally sufficient for an ally library prep. So bivalve, a bit more tricky. So here we would cryoprep the sample. We go for the automated magatract, and we would hope to have enough DNA for an ally library prep. However, we do find that sometimes they fail. In that case, we switch to the manual mollusk nanobind extraction method. So we find that we have around a 50% failure rate from the magatract, and of those we can get another maybe 50% through using the nanobind extraction method. Fungi, super tricky. We actually use the plant bead beating method now for fungi, and the automated plant magatract, because the disruption of fungi is extremely challenging. Also, we find that the DNA after extraction doesn't always sequence well, and actually using the ULI library prep method has shown a lot more success. So we use that both for yield issues and also because we find the native DNA is a bit difficult to sequence. And then an orchid sample. So an orchid here is an example of a challenging plant to sequence, and I will say that not all orchids are going through beautifully just now. But difficult to extract plants go through our plant organic extraction method. We have found that without this extraction method, we do sometimes see failures actually at all points in the process. So moving on to the organic extraction method has been really helpful. And then most recently, we've been processing some coral samples under our ASG project. And for those, there's a little asterisk at the top. We have been bead beating coral, particularly hard coral, but we need to do it slightly differently to how we do plants. So we use different beads and we use different containers. That protocol is in the works. And then depending on how things go, we'll do a manual magatract, and depending on the yield, we might go for a ULI prep or an ally prep. So then after all of that work is done, we do a lot of sequencing. So here is a representation of the kind of scale that we've been enabled to work at so far. This actually goes up to around the end of last year, and currently I think James and his team are sequencing around 60 Revio cells per week, so we have a really high throughput happening. You can see the dots in blue excuse me, are Revio sequencing events, and in green they are seqWell. And you can see there's lots of variation in yields. We have some great yielding cells, we have some that still fail, and you know, it's always a work in progress. We are always evolving, always learning, and always trying to improve methods. But in order to scale, we've had to push some boundaries. So here we see the representation of the CCS yield as plotted against the N50. And you can see it's not strictly, one is not strictly dependent on the other. So it's not a completely linear line. And this has actually been really important to us. And you may ask why. Well, we don't always have the luxury of creating a perfect library. Quite often, we're having to really balance what's possible and what's achievable with the very precious samples that we are working with. So this is some work that Amy has done on some chordates. So these are really precious samples that were actually collected from animals that had sadly been stranded. We have a very small amount of material, they can't be recollected, and we really want to be able to achieve that reference level genome assembly. So for this extraction, Amy's used cryoprep and a manual magtrack version 2. You can see, looking at this femto profile, this is not a good extraction. We've got the peak of the femto trace there is 9.2 kilobases. It's very low. This is definitely non-ideal. However, this was our only option, so we decided not to shear this DNA, but to put it directly into library prep. And then here you can see the outcome of that work. So we've had 4 sequencing events, 4 cells of sequencing. This is on the sequel 2. And you can see that the yield, not ideal, but not bad. So we've got 20 gig, 19, 19, 23. The genome is complete. So even with a non-ideal sample, even having to sacrifice some of those most perfect processes, we are able to achieve these reference level genome assemblies. And actually, this was true for the whole collection. So I think there were around 10 in this collection of samples. We then have some other non-ideal samples. So jellyfish. They've been extremely challenging for us. We started with our standard extraction process and found that the yield was basically non-existent. So we went through lots of different approaches, and eventually, Amy's work again, we arrived on the Amiga biotech extraction protocol and did some very careful selection of the actual tissue that was used for extraction. So again, you can see the DNA extraction on the left-hand side there, not ideal. There's 200 milligrams of tissue have gone into that extraction. That's a lot of tissue to go into an extraction for us. The yield is only 1,500 nanograms, and it's a very kind of spiky profile. Once processed, we've got 600 nanograms of DNA. However, sequencing on the Revio, we yielded 89 gig of CCS, and again, completed the genome. So I think it's been very important for us to push those boundaries, because samples that seemed impossible are actually possible. Okay, so it also requires specialist knowledge. So here is that same data set, but this time it's split between chordates, arthropods, other animals, plants, and fungi and protists. And you can see, again, there's not a strong correlation. The yield is very variable. We do see lots of great sequencing. We also see some failures. So challenging species do remain. So within our extraction laboratory team, we have different specialists who focus in on the failures that we see to try and turn those around. So, for example, Adam leads on other metazoa, and this is an example of a nudibranch sea slug. We often find that slugs and actually marine organisms are quite challenging to extract. And so this particular species went through the standard megatract extraction process, but the low yield that we saw prompted some R&D work. And here, that is one of the examples where we moved on to the nanobind protocol. And here we're able to see the difference between those two extractions. So on the left is the manual megatract femtotrace, and on the right is the trace from the nanobind. And you can see this huge spike here in the ultra high molecular weight zone. And the yield, of course, has gone from 45 nanograms to about 10 micrograms. So this is a huge improvement that we're seeing here. Still just see 50 gig on a Revio cell from this example, so that's still a little on the low side for us. However, it's, this species is now data complete, so we've handed it over to the assembly team. So plants, here's an example of this plant organic extraction method that I highlighted just at the beginning. So here on the left-hand side, we see this particular species, the initial extraction, again, quite a low yield and not very good quality, and that's from a cryo-prepped plant material gone through that plant megatract extraction process. Whereas on the right-hand side, the same individual, the same species, cryo-prepped again, but with the plant organic extraction, the yield is vastly increased. And the profile is pretty ideal. And actually, for the plant organic extraction, we quite often see femtotraces like this. It's a very strong method. So if anyone's working on plants, I strongly recommend that. And then another example, this is a particularly precious sample. This species is critically endangered, and it's only found in Great Orme headlands of northern Wales. And it was submitted to us. Again, on the left-hand side, you can see the results from bead beating and plant megatract. It's not a terrible profile, but it's not ideal. And actually, when it went forward to sequencing, we found that it did not sequence well at all. So Ben, actually, who leads on plants, not only brought in the plant organic extraction protocol, but upstream of extraction, he introduced this sorbitol washing. So this is another kind of module that we're aiming to add onto the protocols that we've published on Protocols.io. For particularly recalcitrant plants, we would recommend bead beating followed by this sorbitol wash and then straight into the plant organic extraction. And then here you can see the profile of the sequencing. And again, so here, this is a really good yield for us. 96.8 gig from a Revio cell is really good. So you'll notice that even for our best examples, we're not quite up there for the Olympics. We're not with the model organisms, but this is a great one for us. So fungi. So the example from Priyanka's work for fungi is actually a lichen. And you can see on the left-hand side that working with power mash input tissue, the yield is extremely low, 13 nanograms, and not great quality. So I think it's just too tough. The power mashing was not opening those cells, not releasing the DNA at all. So then moving on to the bead beating, you can see the femtotrace of that in the middle there. 24 milligrams input, we've got 599 nanograms. So it's still a pretty low yield, but that's enough for us to think about a ULI prep. And that's what you can see here. So this is the output after going through one of those G tubes. Fragment size, 8.5, so it's a little bit short. However, when we sequence this, even for ULI, we were able to achieve 1,000x coverage for the fungal host and then 150x coverage for the algal cobiont. So this is a fantastic result for us, and it shows a lot of promise for this ASG project, where we're looking to sequence host and symbiont in one go. And actually, side note, the high C also separated these two, so you could really see on the high C profile those two different organisms. Okay, and then on the aquatic symbiosis project, I thought I would include this because it's been an absolute game changer for us. Anyone who's working on sponges, we did a lot of work to try and extract good high molecular weight DNA from sponges. And again, this is a symbiosis project, so we're not only looking for the sponge, we're looking for all of these cobionts. With sponges, they're often bacterial. So you can see on the left-hand side, again, we've got the before and after. So there's quite a lot of DNA on that extraction on the left. However, I suspect most of that is bacterial. And then on the right-hand side, we now start to see a good peak in the ultra-high molecular weight zone, and we've got a lot higher yield from this extraction. So this was an extraction after having done the squeeze method. So the squeeze method is published. If anyone needs a link, I'm happy to send it. The sponge material is very carefully washed to kind of move the cells out of the structure of the sponge. And then that is pelleted, and then it just goes into a normal DNA extraction to reveal this yield. But we also find that same prep can be used upstream of high C. So we now have quite a lot of sponges that are going through and being completed, which is fantastic. Okay, and then this is our newest method on the blog. It's actually come from our faculty group. Chris Laumer, Erna King, Vitek, and Adam have been working on bringing this into the core laboratory. So this, Chris has christened it PIMS, picogram input multimodal sequencing. So actually using this method, you can achieve gDNA, cDNA from the same organism. At the moment, we're just concentrating on the gDNA side. We've managed to successfully apply this to nematodes, tardigrades, rotifers, and some flatworms. And actually, tardigrades are the ones that have been starting to go through relatively regularly. So we have around 30 of those that are now sequenced genomes. And you can see the genome scope profile here on the right-hand side. Even with such a tiny amount of DNA, the genome assemblies are looking really good. Also, our colleagues in scientific operation have developed an ultra low high C method for these. So we're hoping that they will couple up beautifully in the not too distant future as work in our program goes more and more towards those really, really small organisms. Okay, so I mentioned pushing boundaries. So another boundary here is the loading concentration, the OPLC. So here again, we've got the yield on the left-hand side plotted against the loading concentration. And again, blue is Revio and green is seqWell2. So what you can most interestingly see here is that on the left-hand side, sometimes at that lower loading concentration, again, we can achieve really fantastic yields. So it's not 100% necessary to load up at that 250, although you can see from the plot here that that's what we do where possible. So again, why? Well, here are a couple of examples. These are slightly older examples, so I think they were sequenced on the seqWell2 rather than the Revio. But here you can see this white pinion-spotted Lepidoptera here. 27 gig of CCS yield from a seqWell2E cell is really incredible for us. That's a very good sequencing sample. But the predicted OPLC at 70% recovery was 27 picomolar. So you can see that's a pretty incredible yield that we saw there. And that is now data complete, so we've handed that over to our colleagues in assembly. And similarly here for this bird species, 29 gig CCS yield, and the OPLC there was at 50% recovery, would have been 22 picomolar. So again, really pushing those boundaries, but actually sometimes works out. I will say it doesn't always work. Of course, I've picked the examples that are lovely examples, but yeah, it's important to know that it's worth trying. And then isopods are an interesting example. We have seen huge challenges with isopods. They have relatively large genomes and quite small body size and quite low yield of DNA. So here we've managed to tackle these species by splitting our DNA extractions and aiming for both a ULI and an LI library prep. So you can see here on the bottom, the top run is a low input library. These have all been run on the Revio. So we've got 27 gig from a low input library, and then we've managed to do 2 runs from a ULI, 21 and 18 gig each. So this is now data complete, which is fantastic, and it just enables us to really optimize the amount of DNA that we're able to extract. Okay, so I may be not sure how I'm doing for time. Oh gosh, I've gone very fast then. Okay, well, I've gone quite fast, but hopefully there'll be some questions. So I will say, all of that having been said, how this is really achieved in the Tree of Life is through collaboration. So I've got a picture of our team here, another link to those protocols. We would love for people in the community to use those protocols and to give us feedback. For us, we find them very useful, but we would love to know how useful they are for other people, and also to share more and more protocols as we develop them. The other thing that we do is hold monthly R&D meetings, which are open to anyone who would wish to attend. So they're on a kind of 6-monthly loop. It's a different taxonomic group each month. So we'll maybe work on fungi one month, and then the next month will be plants, and it goes through like that. If anybody is interested in joining those meetings, they're on Zoom, they're completely open, anybody can contribute. We give an update, like you can see on the left-hand side there, so we talk about our DNA extraction results. We talk about the good ones. We also are very happy to show the bad ones. We'll let people know what's not working for us as well as what is working for us. And we go all the way through all of the different processes that I've shown today. So the fragmentation, the sequencing yields, all of that we're very happy to share with people. Also, if you're interested but you can't come, we do record those meetings, so we can share the recordings afterwards and you can watch them at your convenience. Okay, so then, it takes a village. We have a huge number of collaborators just on the Darwin Project, Botanic Gardens Edinburgh and Kew, the Natural History Museum. I won't read out all of these, don't worry. But we have an amazing number of collectors and really engaged naturalists who are really experts in their taxonomic area. We have lots of teams within the Tree of Life. We have sample management, core lab, assembly, grit, and the faculty teams. And we also have the Sanger core facilities. And anyone who's not aware, we have James and Craig in the audience who are from our Sanger core facilities. They actually do all of the sequencing. And we are especially grateful to them for all of their hard work and their teams, and also for the R&D team in that area of the Institute as well. And to all of the collectors and collaborators in our various other projects that we're involved in. Okay, so I think that's me. Thank you very much, everyone. I hope it's been interesting.